Welcome to Phase Space Invaders. It's your host, Miłosz Wieczór here. Today we're releasing episode 23, and my guest is Zoe Cournia, Director of Research at the Biomedical Research Foundation at Academy of Athens. Zoe's main field of research is computer-aided drug design, and her work these days focuses on developing small molecule anti-cancer drugs that can target specific cancer mutations. Besides having claimed an impressive list of awards and recognitions, Zoe also works as an academic editor for JCIM, the Journal of Chemical Information and Modeling, and used her entrepreneurial spirit to found a mobile app company focused on evidence-based labeling of chemicals in food. Just to give you an idea of how many successful scientific adventures she's embarked on. So we start with a general discussion of the current status and promises of drug design, a field where many things seem trivial but nothing is really simple. Zoe talks about the challenges that precision medicine is facing and how it fits into the grand landscape of future therapies. We go through some success stories, and I ask Zoe how the recent additions to drug design workflows help them in everyday scientific practice, both in terms of physics-based and data-driven models. We then spend a moment talking about the opportunities and responsibilities of her journal editor, and what are the lessons Zoe learned from creating her startup. As always, there's a ton of interesting insights coming from my guests, so hope you'll enjoy the conversation. Zoe Cournia, welcome to the podcast.
Thank you very much for inviting me, for giving me the opportunity to discuss today about computer-aided drug design and related topics. And congratulations on launching this effort.
Oh, thanks. Hope it's useful for the community. So, Zoe, I've talked here before with quite a few people who sometimes venture into drug design or drug discovery just as a subset of what they routinely do. But I think you were the first guest who primarily works on drug discovery, and you have done so for a good while with quite remarkable results. So perhaps at the risk of going into a territory where everyone has a strong opinion already, would you mind sharing your own perspective of where the computational side of drug design stands and what is the biggest promise and challenge of the field as of now?
Yeah, thanks for the question. Of course, everybody's entitled to their opinion, and everybody has their opinions, strong or not. So I'm just going to speak about myself today. I hope nobody will be offended. So I work not in general in drug discovery, but in computer-aided drug design. So that's a specific part of drug discovery because, as you know, that drug discovery has so many stages up to the preclinical stage that is very difficult to work on all of them. So, in my opinion, we are experiencing really a transformation of computer-aided drug design. And that's driven by new softwares, by new methods that are being developed, and molecular simulation techniques. And of course, we have artificial intelligence. While it's not new, it's not a new method, we know that artificial intelligence techniques exist since the 50s. The Nobel Prize in Physics was awarded on that, as well as in chemistry. But although these are not new techniques, we now have more data that we can harness and we can apply these methods. And of course, we also have advances in high-performance computing. So we have more powerful computers, we have GPUs. Quantum computing is starting to pick up. It's not there yet, but we hope that it will be there soon. So there are some promises of computer-aided drug discovery that I can discuss. For example, because of these advances that I just mentioned, it's now possible to screen billions of compounds virtually, which, if you can post-process the results in a rational way, can lead to speeding up the drug discovery process compared to traditional wet lab experiments. We also have the predictive models that I discussed. For example, with artificial intelligence, we have reliable data sets, for example, protein-ligand interactions, bioactivity data, that can enable accurate predictions of drug properties, binding affinities, toxicity, ADME. profiles, and so on. We also have in personalized medicine. We know that this is a new era of medicine. It's called precision medicine. So with computational approaches, we can integrate many different omics data, genomic, proteomics, to design drugs. We can find better targets through these processes. And through specialized computational techniques, we can design molecules for specific mutants of these targets. We also have, people are now starting to work a lot on novel drug modalities. For example, protein degraders, molecular glues, RNA-based drugs, in general, novel therapeutics. We'll discuss a little more next. So that's quite exciting for me because, for example, PROTACs is something that is recently emerging.
Can you introduce the concept to those of the listeners who maybe haven't heard of them, the PROTACs?
So PROTACs is a new drug modality. So basically, PROTAC stands for proteolysis targeting chimera. It's a molecule that is being attached on a ubiquitin ligases. So one part is engaging an E3 ubiquitin ligase, and another part of the same molecule binds to a target protein. So basically, what is
you conjugate two binders?
Yes,
two separate
basically,
targets.
you take up the ubiquitin ligase, takes the proteins for degradation. So you attach one part of the molecule, this proteolysis targeting chimera or PROTAC, onto the ligase, and the other part you target and the protein which you want to degrade. For example, a mutant in cancer.
Mm-hmm. Yes.
And when PROTAC binds onto these two proteins, it's signaled for degradation. Yes.
Thanks.
Also, we have, more structural insights. Cryo-EM has experienced a renaissance lately. So we have more structural data. We have molecular dynamic simulations that are advancing their force fields, QMMM methods, and so on. And also in lead optimization, we have different free energy methods that can be applied for lead optimization that are quite robust. So because now I'm going into the challenges. So for example, in my opinion, one of the biggest challenges in drug discovery is the lead optimization phase. It is the most costly phase. And obviously, optimization for binding affinity in terms of free energy of binding of a small molecule to a protein is very challenging, as well as the ADME profile. So in my opinion, this is the biggest challenge. And we have tools. For example, free energy perturbation calculations can be used on a congeneric series of molecules, so very similar lead series. With that method, you know which one is preferred for binding to the target protein. However, these calculations are very costly, so they need computational power. Also, molecular dynamic simulations need computational power. QM calculations remain resource intensive. So while we have seen an advance in computational power, we still need more. Who knows, perhaps with a quantum computer.
It makes sense that lead optimization is among the most challenging, right? Because screening you can do with very cheap.
It's a bottleneck.
You can do with very cheap scoring functions, but here you have to employ those physical, very kind of precise methods to see all the influences of small functional groups.
Exactly. And I was about to say, so the next challenge is the accuracy of the simulations, also of docking, scoring functions. They are fast, but they're not so accurate. So this is a challenge. It's good enough to find a hit, but when it goes into lead optimization, you need more accurate simulations. And of course, that also goes into force field development, into going into more complex biological systems. For example, even in lead optimization, you have an accurate method. Let's say free energy perturbation calculations, you have a good crystal structure, you do the calculation and it fails. It doesn't give the experimental result. Why? Maybe the parameters of the force field are not good, or maybe there's a water molecule that entered and is stable in that pocket that doesn't allow for the full complexity of the system to be simulated. So there's the complexity of the biological system is another challenge that remains, including protein flexibility, solvent effects, allosteric regulation. Mm-hmm. Another challenge is the uncharted chemical space. As I mentioned, we are able now to screen billions of molecules, but the vast chemical space has not been exploited fully, in my opinion. So going beyond non-scaffolds is a challenge. Perhaps artificial intelligence could help in that respect. And since I mentioned artificial intelligence, I could also speak about some challenges being faced there. One of the biggest challenges that we are also facing in my lab is data quality and bias. We have data, as I mentioned in the beginning, but we don't always have the data that is needed to make an efficient algorithm and robust algorithm. So data quality and the bias is one of the biggest challenges right now. Basically, in machine learning and artificial intelligence, what you put in is what you get out. So garbage in, garbage out. If your data set, it all depends on the data set. If the data is not clean, if the data is not unbiased, and if the data is not trustworthy, you are not going to get trustworthy result. So poorly curated or biased data can lead to misleading predictions. And for me, that's one of the biggest bottlenecks right now with artificial intelligence and drug discovery.
So it's also consistency between different data sets, right? Because maybe one method is used in several data sets, but the details will make those results uncomparable.
And you also asked me about future directions, but before I go into that, I would also like to say that for now, AI is more like a black box. It's difficult to understand why specific predictions are made. And also, in my opinion, they don't provide physical insights. So just to conclude this challenges part, I don't think that molecular simulations or physics-based methods are going to be replaced by artificial intelligence, simply because in artificial intelligence, you mostly interpolate. You don't really extrapolate, and you cannot make new insights or predictions. You only post-process what the knowledge that you already have. You don't create new knowledge. So in my opinion, physics-based simulations will still continue to provide new insights and be used by our community.
Right. I think I've seen this commentary recently about this diffusion-based methods, right? That they mostly memorize the training set and then give results that are similar to what was in the training set.
Exactly. And this is our experience. We have tried multiple flavors of AI, and it's always the same. If the training set has not seen a property, it cannot reproduce it and it cannot predict it.
So we need explainability.
Yes, exactly.
Right. So let's jump then into one of your specialties, which is precision medicine through allostery. If you care to explain the concept, what has been proven to work? What are the things that you hope will work soon? Where is this subfield now?
In my opinion, it's a very exciting field, and it's a future of drug discovery, because allostery leverages this concept of allosteric regulation to develop new molecules that are specific, even isoform specific within a protein family. And they can be also mutant specific, so they can be targeted to a single point mutant over a wild type of a protein. And these modulators that can be developed through allostery, they can be used for targeted therapies to treat individual patient profiles. So let me explain what is allostery. To my understanding, allostery refers to the process by which a molecule would bind on one side of a protein, which is called the allosteric site, and then it uses a conformational change or a functional change at a distant site, the orthosteric site. So you have the active site, and then on the surface of the protein, you have other places which can influence the orthosteric site or the active site by modulating a far site. So this approach provides unique advantages because it's very specific, it's very controlled, and addresses challenges by traditional active site inhibitors. So, for example, I can give you some advantages that are in my mind. So these allosteric sites, they are less conserved across protein families. For example, if you have protein kinase A and protein kinase B, and they're very similar, they're doing the same reaction, the active site is very similar. But then these allosteric sites, they will not be similar across the same protein family. And that allows for creating modulators with greater specificity and reduced off-target effects. Another important point is that these allosteric modulators can fine-tune the protein activity. For example, you can achieve partial inhibition instead of complete inhibition. Or they can be targeting specific patient mutant profiles. Also, there was a study, several studies actually in the last 5 to 7 years that show that allosteric modulators are less susceptible to resistance mutation compared to allosteric drugs. So we all know the problem in cancer of drug resistance. You take one drug, and then after a while, the cancer mutates and avoids this drug. Now, people have shown that if you treat combination therapies, the orthosteric inhibitor together with an allosteric inhibitor, the resistance profile is eliminated or limited. And that's very important.
Mm-hmm.
And
do we know why, by the way?
I mean,
or is it just
exactly? And this concept has not really been exploited so much. So I think with allostery, we will have a new era in drug discovery. And for sure, we can exploit all targets with new chemical space because of these new pockets. And also we can target proteins that have been considered undruggable in the past. And I will give you examples because you'll ask me for examples. The most famous or recent example is the example of KRAS. It's a protein that's mutated in 30% of cancers. And it was not possible to drug it because there was one pocket, the orthosteric site, which had a picomolar substrate, GTP. So you couldn't really remove it because, it's a picomolar, so nothing is better than that, obviously. But researchers found, Kevin Shokat and his lab, they found an allosteric pocket where they designed sotorasib. And this sotorasib targets a specific mutant in cancer, and it was not possible to do it before. Previously, KRAS was undruggable. It was not possible to target it. But with KRAS, allostery, it was possible to launch a drug, an FDA-approved drug, in the market in May 2021. So I think we're experiencing, I have more examples, but I'm sure we have more items to go.
Well, yeah, it's great to know that there are a few at least. So in this case, this doesn't rely on any mutation, right? So it's kind of when the patient has a mutation,
yes.
No matter what the mutation is, it will work.
No. This is a specific mutant.
Or it relies on
it's a specific drug targeting the glycine 12, which becomes a cysteine. So it's a G12C specific mutant, and that goes back to the concept of precision medicine. So with allostery, you can really target specific mutants. So for the other mutation, G12D of KRAS could be another mutant.
That was my next question. So how much can we rely on these repetitive mutations? How many cancers really feature a small set of well-known mutations that are always the same, right? Rather than, maybe thousands of small mutations that never repeat.
Yeah. Let me just finish the last question by saying that we have now a network, a European network that is called Allostery in Drug Discovery. And 11 companies and 13 academic partners are participating in this consortium. We are having our annual conference in April 2025 in Athens. So if you type Allostery Drug Discovery Conference on Google, you will find that it's between 9 and 11 April of 2025. I hope people who hear us will join us for the conference.
Okay. Yeah. Participants welcome. Absolutely.
Yeah. So let's go back to the question. What was the next question?
The question was how much are cancers driven by a small set of well-known mutations versus maybe a huge number of random mutations that just mess up with something that we don't really understand.
Okay, this is more a question for a biologist or a medical doctor. I am a chemist, so Because I work at the Biomedical Research Foundation in the Academy of Athens, I have exposure to geneticists and biologists. So I will just give you my uneducated experience here. So, yes, I mean, cancer, because I think the question is on cancer. Cancer is thousands of diseases, because often I get the question, when will we find the cure for cancer? Well, cancer is not one disease, it's thousands of diseases. So you have different subtypes. Breast cancer, for example, has over 100 different subtypes that we have identified. So it is a multiple diseases. It's not just one disease. And in the specific types of cancers, there are obviously efforts going on. For example, the KRAS mutants that we discussed, EGFR in non-small cell lung cancer, BRAF in melanoma. We have PI3 kinase again in breast cancer. So roughly, I would say that maybe there are around 100 mutants that are being targeted or in the preclinical stage or in clinical trials or in the market. But this is very far from the actual mutations that exist in cancer. That's why I really think that gene and cell therapies that are coming up, because cancer is a genetic disease. I mean, you can do so much with a small molecule. You can block the disease, but you don't cure it. You never cure it with a small molecule. You need to eliminate the mutation. And this is possible only with a gene therapy. Gene therapies, of course, are not available right now, but they will be. I'm sure we saw the advances on CRISPR, and we have data that show that gene therapy is possible. So you could replace with a viral vector, you could replace a specific gene with a correct gene. That would cure cancer. The small molecules only slow down the progression of the disease, and as new molecules are coming, people are changing their therapies, and they have a better quality of life and a prolonged life, but you are never cured if you don't replace a gene. So I think the future lies in gene therapy as well as cell therapies, the CAR T cells for some indications, a few cancers.
So again, this, for context, these are therapies that rely on ex vivo treatment, yes, if I remember correctly. This CAR T therapies?
The CAR T therapy is for, mostly for immunological cancers. Well, basically, they take your cells and they reprogram them to find the cancer cells and kill them. So then they kill all the cancer cells,
right,
which means that the mutation is not there in your body. So that's the concept in the course discussion of the CAR T therapies. But obviously, these are very costly. I mean, I'm not sure if the number is still correct, but I heard it's like $1 million to get a therapy.
Right. So that's part of the problem here, right? That sometimes we will have those almost bespoke therapies that will be made for a single patient, but then that means that a team of scientists is working on an individual case.
Exactly.
Do we also have effort in this like orphan disease direction? Let's say those very rare diseases that can be maybe targeted by repurposing, or this is something that has less interest in the field?
Obviously, I mean, specific efforts are being made for the orphan diseases. And of course, repurposing is one way to find new molecules using old modalities. And of course, yes, experimental repurposing of drugs, it's something that's going on. And we saw it a lot with COVID. It is an approach that can be done.
All right. So we probably need hundreds and thousands of scientists working on every possible branch of this problem, right? With so many mutations, so many separate diseases that rely on individual mutations, and maybe each of them requiring some particular designer treatment. That's really the challenge of the whole field. So if you don't mind sharing with us, how does your typical workflow look like? If you approach and use, let's say, a new system, a new mutation, how would you approach this problem now versus maybe 10 or 15 years ago?
Yeah.
Probably most of the people take their knowledge from.
Yeah. In our lab, a typical workflow would start with allosteric or cryptic pocket identification. So in the beginning, we need to find a new pocket if we don't have a pocket. But usually we work with challenging targets. So that means that the pocket is not there or it's occupied by a high affinity substrate. So the first thing to do is to find the pocket. And how do we do this? We run typically molecular dynamic simulations, either vanilla or unbiased simulations, or biased simulations if we know that the conformational change is happening. We bias the energy of the system towards this direction, towards a big conformational change. And that conformational change in the process reveals pockets that were unseen before, and also reveals how the protein is allosterically modulated. So we screen with different tools for pockets initially, and then we test whether these pockets are allosteric. What does it mean? It means we want to see whether the pocket that we have discovered communicates with the active site or other functional areas of the protein. In other words, whether there are networks connecting the two pockets. And there are a number of ways to do that, usually with cross-correlation of residues, normal mode analysis. So there's many tools out there. So once we decide that, okay, this is the pocket of interest, and, we check whether the pocket has available druggability, if it has good characteristics that would make the pocket druggable. We usually would run some simulations with a probe in the pocket to see how it evolves in time, whether there will be an opening or closing. And we are also using machine learning techniques for understanding these allosterine proteins and for predicting if a pocket will be allosteric through data sets that we have gathered from the literature. So this is a project currently running in our lab. So identifying allostery through machine learning methods. So that's the first part, to identify where is an allosteric pocket. So once we have the pocket, or it could be the active site, in some cases the active site is still interesting, then we proceed with a normal virtual screening workflow. And although, I say normal, and one could say, okay, virtual screening, anybody can do virtual screening. In my experience, I see that, not anyone can do the virtual screening. You need deep chemical insights to understand the protein ligand interactions of what it would possibly be in reality and what you see on the screen. So you need to know your chemistry quite well. You need to know the hybridizations. You need to know whether a molecule would be cis or trans configurations, whether it would be an EZ ester, the hydration that you see if you choose to put molecules in docking. So there are a lot of parameters that are crucial there. So a good virtual screening exercise would mean that the person knows what they're doing. And of course, we also have the ensemble-based method, because in virtual screening, the target is not moving. You can have multiple structures to virtual screen and then get the consensus results from those structures. So this is the classic docking. In our hands, it has success rates of around 20%. So if we screen millions of molecules, we select 100. Usually between 10 or 20 molecules will have micromolar affinities. And then if you have micromolar affinities, you go into lead optimization. Lead optimization, you can do it with de novo design, having ligand growing programs that would grow in different directions your initial molecule, and then calculate the free energy of binding either with docking again, or if it's sufficiently different, the molecule, or with free energy perturbation calculations, or you can do scans with free energy perturbation methods. And lately, of course, we also use AI-driven generative models. So these create novel molecular structures. Although we have seen, for example, diffdoc is one method, but we have seen that they are not always there yet. Sometimes the molecules that are being predicted by these generative AI tools, they don't have the correct druggability profile of a molecule. So sometimes the groups that appear are far from those that exist in drugs that are currently on the market. So basically after that, when we identify a hit. We verify it in vitro testing. We run lead optimization cycles. Then again, iteratively going into experiments. And in the meantime, we also do molecular dynamic simulations. We have found molecular dynamic simulations to be a good predictor of the dynamic behavior of a molecule and whether it will be stable in the pocket. So we're routinely, I would say, we also do molecular dynamic simulations on the protein ligand binding. And something that is crucial on this step is the reproducibility. So you cannot do it only once and see it's stable, so I'm fine. You have to do multiple replicas. I would say 10, ideally. But if you don't have the resources for 10, 3 are good. So I would say this is the workflow that we follow.
I'm curious, when you mention running simulations with the ligands, are the new neural network potential something that is going to help here? Or because I know that many of those strange or new molecules don't really parameterize well or require huge effort to parameterize.
Yeah, that's a really big issue. So every molecule is unique. It has unique chemical properties. So the parameters are not transferable directly from the force field because, okay, the amino acids have been very well parameterized, the lipids have been very well parameterized, the water, but these don't change. They're always the same. The small molecules are always different. So efforts in the parameterization of small molecule is needed before you run this relation. And we have a history in force field optimization. So we do take care of that part. So in my opinion, when they are ready and robust, the new machine learning potentials, they will definitely tremendously help speed up the calculation. Because, okay, getting the parameters, we can also get them, manually, but this takes a lot of time. So if we could have them in a matter of seconds, this would be a game changer.
Yeah, we had Alex McCarroll on the podcast, who is one of the main people behind Charm Generalized Force Field, and that's probably one of the go-to places to get parameters, right? But they also give you those scores saying, oh, this is very unreliable. You have to validate separately.
Exactly.
So it's never that easy.
Exactly.
And then my other question was, how do you then, once you're done with lead optimization, are you also incorporating this ADMET, so exactly absorption, and distribution, metabolism, toxicity, excretion data into your
absolutely. I mean, that's critical. That's a critical part of a practical approach to drug discovery. So these ADMET properties, they can determine whether compound can become a viable drug. So one cannot overlook them. Binding affinity is, of course, very crucial parameter in drug design, but ADME is the one to determine whether a compound can become a drug. So these properties have to be integrated in the drug design pipeline, both through predictive models and experimental validation. So we have not worked with creating predictive machine learning model, although the data, I would say, is there. It's possible to do it. And of course, there are tools, DeepChem, ADMET Labs, Swiss ADME. There are many tools out there that we use. And you can calculate the half-life, permeability, hepatotoxicity, and other properties also that we calculate that done in machine learning, like molecular weight and solubility log P. So we definitely take a look at those. So far, my experience is that also these models are not there yet, but they are improving, especially the commercial solutions, the ones that you pay for. They are reaching a very good level of validity. So I would say that's definitely a critical task in drug discovery.
It feels like drug discovery is one of those fields that are kind of driven by big capital, maybe more than on the methods development side. Not to say that Google isn't working on method development with AlphaFold and things like that. The big money is probably exactly in the drug discovery, drug design side. And to move away from the drug design side a bit, what are your experiences as the academic editor of JCIM, the Journal of Chemical Information Modeling, and also as an entrepreneur, right? This is also something you do. How are those sides of the, let's say, academic enterprise different from the standard life of a PI? If you can share a few
absolutely.
insights or experiences.
First of all, the editorial work, I'm really grateful for the trust of our editor-in-chief, Kenny Mertz. I found it extremely exciting. I learned so much, but I could also contribute a few things that I think are important. For example, data reproducibility, principles, open science, advancing women in science, especially in computational chemistry, some practical considerations of doing molecular dynamic simulations, also machine learning. So a very big part that I find rewarding, apart from the typical assigning manuscripts, reviewing manuscripts, and so on, is the policy making that you can do. And we have published a few editorials. For example, one is on method and data sharing and reproducibility of scientific results. And I think that JCIM is a pioneer in this area because before we accept a paper to go for review, we ask the authors to provide all the data that were used to generate the results. Parameters, topology files, scripts, analysis scripts, the data sets for training. Everything that is needed to reproduce the study should be part of the supporting information or should be uploaded in an open source repository. That's really crucial, in my opinion, because the era of data available upon request is hopefully behind us. Is we need to make how many billions of euros are spent in duplicating research efforts because the data is not there. So every year. So we need to go over this. And that's why I'm really grateful to the vision of Kenny Mertz, who enabled this policy in JCIM. Now all the data from all the papers that are needed to reproduce the data in the paper are available. We don't ask for the outputs or trajectories. Okay, this is on the person if they want to make them available. But to generate the data, all the inputs need to be there. So that was one really important contribution for me. So open data and open science. And in terms of women in science, we also published 2 special issues, which for me, there are 2 sides of the coin. One says, why are women special? Why should you have a special issue on women in computational chemistry? But I think, it has been shown that there are inequalities. So we really thought that we could publish this special issue to build support networks, to make aware of the issues that are there, so that we promote more, for example, women speakers, moderators in conferences and panels, to ensure that gender balance on important decision-making committees is there. If we witness unfair comments, we are all saying something about it, recognizing implicit bias. For example, to ask our governments to offer funding for professional reentry after a woman gives birth. There are so many things that feel where we have some work to do that I thought it was important.
It feels that the computational side was always particularly biased towards men. So that's a welcome.
And that's what the statistics say. I don't say this, but it's true. Because if you see the statistics, when you are at a PhD level, you are 50-50. 50% male, 50% female. But when you go into the professor rank, 26 professors is women, and 80% or 74 is male. So clearly there's something going on there. And I think it has to do a lot with when you are about to become a professor. This is on your childbearing years. You want to have children, you want to grow your family, and typically this stalls you from being promoted to a high rank.
Right. We just had this conversation with Lucy.
Exactly.
Who has just become a mother last year. And she made this point that once you have tenure, it's really easy, but then biologically it can be also hard to meet those goals.
Yeah, absolutely. And we don't count also that because, when we are in the academic track, we are striving very hard to go close to the tenure in any, not only in academic track, but also in the professional industrial track. And you may be unsuccessful in creating a family. You may have miscarriages, infertility, unsuccessful adoptions, and these take a big toll on the women's psychology, and these stall the progress. So that's why I think we have low percentages of women in high professional ranks. But with these special issues, what we hope to do was to create a network. So we asked all the authors that were contributing to this special issue to write that they support advancing women in science. In this way, if one opens this issue, they know that, okay, these authors are supportive. So I could ask them to mentor me, for example. So this is what we wanted to build with these two special issues, to find people who are believers of advancing not only women, but in general, to promote equality in all aspects, and to build a network of these people that are believers in equalities. So that was very important for me. So this policy making, let's say, also for some guidelines or best practices for molecular dynamic simulations. I mentioned it before. I think reproducibility is key. So we published this editorial on best practices on performing molecular dynamic simulations, where we say that, okay, look, you have to do a triplicates of your simulation. One simulation is not enough. So that was important for me. And apart from those policy-making stuff that I mentioned, there's also some other insights for me personally. So I learned that it's important in an article to have clarity and focus, to show the novelty. Often the comments I receive from the reviewers is that the manuscript is not novel. But it was not that it was not novel. The manuscript was novel, but the authors did not pronounce the novelty enough to be understood by the reviewer. So that's important, to show the novelty of the work, to validate the work, to make conclusions that are supported by the data. And also I learned how to provide constructive feedback, because what's important is to make a scientific contribution to the community that is valid and will be used. So we need to provide reviews that are constructive, are respectful and solution-oriented. And we already talked about the data. That's another important point, data sharing and transparency that enables our reproducibility and open science. And yes, I think I already said a lot about that. I found it really rewarding. I learned a lot of things, also about writing my own manuscripts. But I also learned that, the editors, their job is really to facilitate that new innovations are coming out into the scientific community that are valid, and that they don't have plagiarized content, or that the discoveries are solid and have novelty. So that is like a pre-screening tool for the community.
Right. Out of curiosity, how many papers go through your hands every day?
Well, not a lot. It's not every day. I would say maybe 5. Yeah, 5 or less.
I wanted to connect it to the point about highlighting the novelty, right? That if an editor assigns a given number of minutes to every paper, because of course you cannot spend all your day just reading the papers all the time.
Yeah, I mean,
those things have to be highlighted, have to be clearly visible out from the first glimpse, right?
I don't do the review myself. I assign the manuscript reviewers.
Right.
So
you decide whether it's interesting enough, I guess.
Yeah,
right?
I mean,
is it part of the job, or
very few manuscripts?
How much of the job is
few manuscripts that come into JCIM are not going into review. I will give you some examples. If you have a drug design paper that comes in, but there's no experimental validation, this will not go to review because it's just a putative computational hit. Obviously, if it's just computational, we don't have experimental validation. We cannot send it for review because the result is questionable. So only such extreme cases will not go to review. I am not the one to judge the novelty. I think it's useful for the people to hear what the rough rules of thumb are from the editor. Like what I can give you more
goes
examples.
in, what goes out.
For example, if there is a paper coming in that explores a particular imidazole salt and its ionic solvation shell that is very narrow. This goes into physical chemistry. So there is a scope for JCIM. So basically one of the best guideline to remember is that when you are submitting to a particular journal, you have to read the aims and scope of the journal. For example, JCIM, the Journal of Chemical Information and Modeling, is a journal that wants to have advances in applications of molecular simulations, developments in multi-scale modeling, artificial intelligence, machine learning models to chemical and biological data. So there is a list of what we are interested in, QMMM applications and methods. Now, if it is a very narrow system that focuses on the physical chemistry aspect of the system, then the Journal of Physical Chemistry is more appropriate. So people should also read the scope. The author should read the scope of the journal, of each journal.
A good point. Sure.
Yeah, you also asked me about the entrepreneurship part. So we have developed a small startup company. It's called Ingredio. It is a mobile phone application that helps consumers understand dangers from chemicals. So basically, it's a mobile phone application. It's available on Android and iPhones. It's free to download. What it does is that it has a database. It has collected the peer-reviewed literature from PubMed and PubChem. And we have collected also the database of chemicals that are found in food and cosmetics ingredients. So we have created a machine learning algorithm using natural language processing that can identify causal relationships between ingredients of food and cosmetics and adverse effects on human health. So let's say that one product contains one specific chemical. Then the user with the app takes a picture of the bottle. The algorithm determines the chemical ingredients, looks into the database, finds them, and then we assign a specific score of whether this ingredient causes adverse effects on human health based on the peer-reviewed literature, based on scientifically reviewed data. And that comes up to the user as a color-coded score, which says, okay, if an ingredient is red, it means probably it's not good for you, and there's an explanation about it. If an ingredient is green, then it means it's not harmful. And the key insight that I think I got from this process is first that when you're going into an entrepreneurial adventure, what is important is to solve a problem that people have, a real problem. And this idea came from my friends who were alarmed by news that they heard that some foods cause cancer or diabetes, and they couldn't read the label because, okay, they're not chemists. So I could hear all the time that we don't know what we're consuming. So this was a problem that my friends had. And I also had this issue. Although I'm a chemist, I don't know all the E ingredients, E101. I cannot remember it by heart. So we made this application for people to use in the supermarkets right on the go. So it's really important to target a real life problem. And it's the most important thing that I learned is very important is not the money, because money you can find, or if your product is good, you will generate the revenue. It's to get a good team of people that will work with you and to hear your customers, hear what they have to say and what they need and the market need. That's the most important thing. The money is, of course, a bottleneck, but it will be, if your idea and product is good, you will find the money. And there's also a lot of support for SMEs. If European funding opportunities, there's so many incubators these times, because I also am not a business developer, right? I haven't studied finance or economics. So I went into accelerators, incubators, and learned the tricks of the trade.
Right, I see. No, I think it's also a great connection, right, between the academia and the public in this way that our results can somehow be transferred into what the people see and hear.
Yeah, definitely.
Okay, I think we've got a lot of insights from you. Thank you again, Zoe Cournia. Thanks for sharing your experiences and the insights and being on the podcast.
Thank you for inviting me.
And have a great day.
Bye.
Thank you for listening. See you in the next episode of Face Space Invaders.